Cultivating viruses in embryonated bird eggs. Now, an embryonated egg is one which has an embryo developing inside. And embryonated eggs were first used for cultivation of virus by Ernest Goodpasture in 1931. The eggs that are used, they must be sterile. The shell of the egg must be intact and healthy. And normally the eggs that are used for such inoculation include chicken eggs, duck eggs, and turkey eggs. The egg can be used for inoculation between 7 to 12 days after fertilization. An embryonated egg will offer several sites for cultivation of viruses, and the different sites for viral inoculation. What that means is, where are we going to inoculate the virus in that egg? So we can inoculate into the chorioallantoic membrane, or the allantoic cavity, or the amniotic cavity, or into the yolk sac. Now first we need to see what is the stage of development of the egg. And for that we carry out a process called candling an egg. So what this means is that the egg is placed in front of a light source. And when you keep it in front of the light source, you can see through the egg. You can see where are the veins, you can see where is the embryo, you can see where is the air space in the egg. All that can be seen if you just place the egg in front of a source of light. That is known as candling an egg. So before we inoculate the egg, it has to be first prepared for that inoculation. So first all these, the location of all these things is first seen. And then the shell surface has to be disinfected with iodine. Then based on where you want to make a hole in the egg, they make a pencil mark. They'll make a pencil mark there and make a small nick. A small nick is made on the shell, or a drill and a hole is drilled at that region. And using a syringe, then the virus will be inoculated inside. So where exactly should the virus be inoculated? That would depend upon which virus we wish to cultivate. For example, if you look here again, you see here that you will inoculate the virus into the chorioallantoic membrane. When what you want to grow is the pox virus, you will inoculate it into the chorioallantoic membrane. And the inoculation into the allantoic cavity. This is most popular, but it's a simple method. It is employed for the growth and replication of the influenza virus for vaccine production. The amniotic sac is inoculated, especially in the case of duck's eggs, for cultivation of the rabies vaccine, and also for isolation of mumps and influenza virus. And inoculation of the yolk sac, that's the simplest method for growth and multiplication of most mammalian viruses. Now let us take an example. Now suppose we are interested in preparing a vaccine against the influenza virus. For producing 1 microgram dose of vaccine, we will need to cultivate the virus in 2 eggs. So we want the virus to grow in the chorioallantoic membrane. So for that, as said earlier, we have to place the egg in front of a light source to find out where the veins are, where is the allantoic cavity, and so on. So it is, the allantoic cavity is present just below the air space. You can see there's an air space here, and here is the allantoic cavity. Now this area, which can be seen while candling, that area is going to be marked with a pencil, and a small nick will be made. Now here you can see that here the egg is getting punctured here. A small nick is made here by puncturing, and the inoculation of the influenza virus will happen at that point using a 1 ml syringe. Now, so a needle will pass through this hole in the shell. It will pass through the chorioallantoic membrane, and the virus is actually placed in this cavity, that is the allantoic cavity, which has got the allantoic fluid. There'll be a hole now in the shell because a hole was made here while drilling. Hole is through that hole only the virus was inoculated. Now that once the virus has been inoculated, this hole has to be sealed, and that is done with the help of melted paraffin wax. Now this egg, which has been inoculated, it will be incubated at 37 degrees centigrade for 48 hours. So at this time, when it is being incubated, what's going to happen? During the incubation period, the viruses that have been injected into the allantoic fluid, what they'll do is they'll latch on to the chorioallantoic membrane. The cells in that membrane, they will be the host cells for the viruses. So the virus will enter into those cells and start to multiply. So many virus particles will be produced into those cells. The cells will burst and will release the new virus particles into this allantoic fluid. So our next step is we want to collect the virus. We want to harvest that virus. So what do we do then? We break open this top shell after 48 hours, and then using forceps, the shell flap is removed, and the shell membrane and the chorioallantoic membrane are pierced, and with the help of a sterile pipette, the allantoic fluid, which is now having a lot of viruses in it, that is harvested. And then the harvested fluid can be used for vaccine preparation. The harvested viruses can be used for vaccine preparation after isolation and purification. As mentioned earlier, this kind of inoculation can be carried out in bird eggs. But among the bird eggs, the most preferred are the chicken eggs. And why this is so, you can see here. Chicken eggs are inexpensive, they're large cells, they're readily available, easy maintenance, sterile, an ideal substrate. There is no immune interference. The membranes are sensitive to even those viruses that do not cause infection in adult birds. And the membranes provide ideal inoculation sites. Many avian and mammalian viruses can be cultivated, and this method is widely used in vaccine production. Of course, the same advantages also hold good for other embryonated bird eggs, but especially due to the cost, effectiveness, their availability, and easy maintenance, chicken eggs are the most preferred. The disadvantage of using embryonated eggs for viral cultivation is that for growth of different viruses, you need to vary the site for inoculation, and this requires a lot of expertise. So this was information relating to cultivating viruses in embryonated bird eggs. We can also cultivate viruses in vitro that is in cell cultures, which are grown in Petri plates and test tubes and in culture flasks. We will learn about this in the next video. Thank you.